to present work on interpretable deep learning for multi-level models of psychiatric disorders. This is joint work with members of the Gerstein Lab at Yale and was done in association with the NIMH PsychENCODE consortium. Psychiatric disorders have some of the highest heritabilities across all disorders. For instance, as shown on the table, the narrow sense heritability for schizophrenia and bipolar disorder is estimated to be at least 70% based on family studies. By contrast, the common SNP heritability estimates for these disorders are substantially lower, creating a heritability gap. This suggests that these traits are highly polygenic, influenced by common and rare variants with small effect sizes, whose impacts converge at higher levels of organization, such as the neural circuit level. Consequently, while some molecular determinants of these conditions have been discovered, in general, their underlying mechanisms are far from fully characterized. In addition, like any disorder, we expect liability for psychiatric disorders to depend on genetic and environmental factors. We expect these to be mediated by intermediate molecular layers, such as the transcriptome and epigenome, both during and after development. This picture is further complicated in the case of psychiatric conditions, since, as suggested by the schematic, we expect these traits to substantially influence behavior, which in turn feeds back to the environment, leading to a complex genotype-environment-phenotype map. This means that the causal impact mediated by intermediate molecular layers is in general greater than that arising from genetic determinants alone, but less than the total information shared between trait and intermediate layers, which includes a counter-causal component. We develop an interpretable deep learning framework capable of jointly modeling the dependence of a trait on variation at all of these layers. We allow prior knowledge to be embedded in our model, variables to be imputed when missing, and latent factors to be learned. I'll start by describing the outlines of our integrative analysis of the PsychENCODE data. The work I'll present forms part of an integrative capstone paper for PsychENCODE, along with a series of other analyses that I'll only touch on briefly. Our full integrative analysis will be appearing with the developmental and cross-disorder capstone papers as part of the first PsychENCODE publication package in the forthcoming issue of Science. We integrate genetics, transcriptomics, and epigenomics data from several sources, including PsychENCODE, GTEx, and epigenomics roadmap data. The PsychENCODE data contains mainly bulk RNA-seq data from the dorsolateral prefrontal cortex of around 1,800 postmortem subjects, including schizophrenia, bipolar, ASD, and control subjects. A limited amount of epigenomics, high-C, and single-cell level transcriptomics data is also used. From this data, we identify brain-specific genes, modules, and enhancers, deconvolve the bulk expression data into cell type fractions, identify expression QTLs and other QTL types, construct gene regulatory networks, and integrate all of these features in our deep learning framework. Before describing the framework, I'll briefly talk about the eQTL and GRN analyses, gene regulatory network analyses, which form the key components of the model. From the PsychENCODE data, we identify around 2.5 million brain eQTLs, which is substantially higher than previous studies such as Common Mind. We analyze the e-SNPs for enrichment of GWAS variants and show these to be enriched in psychiatric and cognitive level traits. We also use the data to identify chromatin QTLs and cell fraction QTLs. Our GRN analysis builds a network of regulatory relations between transcription factors, enhancers, and target genes. This proceeds in several steps. First, we identify all potential enhancer-promoter links within TAD regions from our high-C data. Then we identify all transcription factor target gene links mediated by transcription factor binding site motifs in associated promoter and enhancer regions. Finally, we prune these links by an elastic net regression which predicts a target gene's expression value from its potential TF regulators. This produces a global network from which we can extract subnets for individual cell types using identified cell type marker genes. I'll now describe how our deep learning framework integrates these levels of structure. We build our model via a series of stages within a Boltzmann machine framework. This slide shows a series of networks in which the inputs are at the bottom and the outputs are at the top. Inputs consist of SNPs, which are always known, which are shown as C, and transcriptome, epigenomic, and other data, which may need to be imputed and is shown as X. We can formulate a series of models, including logistic regression and conditional, restricted, and deep Boltzmann machines, by defining energy functions over these variables and an output trait, such as schizophrenia, labeled Y, along with various levels of hidden variables labeled H. This slide gives a detailed representation of our full model, which we call a deep structured phenotype network. This is based on an expansion of the conditional deep Boltzmann machine energy function. As shown, the intermediate phenotype variables X at level 1 of the network are expanded into 4 sublayers, which include gene expression values, enhancer H3K27AC levels, cell fraction estimates, and co-expression modules estimated using weighted gene co-expression network analysis. As shown in the lower blowout, the connectivity within these sublevels and between the genetic SNP data is used to embed prior knowledge learnt in the GRN and EQTL analyses, respectively. Hence, we introduce lateral connections within and between gene expression and enhancer variables to mirror the regulatory relationships learnt in the GRN, and we use the QTL linkages identified to link the SNP variables to specific sublayers. In addition, the hidden layers can be interpreted as higher order groupings over lower level variables, which generalize the WGCNA modules. The model is parameterized by a series of weight matrices with sparsity induced by the prior structure imposed at particular layers. So this slide summarizes the training and inference in the model. We use variational mean field for approximate inference. During training, we use persistent MCMC to evaluate the gradient, and at test time, we minimize the free energy to perform prediction of intermediate and high-level phenotypes. In our full model, we also include a final training step to optimize the model for feed-forward prediction following imputation. We compared the performance of multiple variations of our model in predicting psychiatric traits from genomics data. In all tests, we created testing and training partitions of the data, which are balanced for controls and cases, as well as covariates including age, gender, ethnicity, and experimental assay. The percentages shown are averages over 10 such data partitions, where chance is at 50%. We observed that the full DSPN model provides a 6-fold improvement in accuracy above a logistic regression using genetic predictors, which provides a baseline polygenic risk score. The full DSPN model also provides a 2.5-fold increase in accuracy above a logistic regression model with access to gene expression data, demonstrating the advantage of using latent factors to learn higher-order structure. Further, the version of our model where we impute the intermediate phenotypes, such as the transcriptome, provides about a 3-fold increase over the baseline polygenic risk score, demonstrating that using the prior structure at these levels helps to identify the genetic component of liability. Finally, we can convert all predictive performances into liability scores, allowing comparison with narrow sense heritability estimates on the same scale. For the last part of the presentation, I'll briefly outline some of the methods we've developed to perform interpretation of hidden units in our integrated model. As shown in the left panel of this slide, the methods I'll describe can be seen as a special case of a general method of multi-level network interpretation, which we call rank projection trees. This method picks out groups of inputs which influence an output non-independently. When applied to the DSPN on the right, this method forms a sparse DAG over the network, including both positive and negative weights, shown as red and blue, which are ranked by their absolute values, shown as full and dotted lines. Given an input node such as A and B, we can find the best positive and negative paths through the network in terms of their combined rank scores, and generate higher order groupings associated with each node on this path by selecting descendants in the DAG, which contribute to either positive or negative variation in these nodes. These higher order groupings, or HOGs, can be treated analogously to gene expression co-expression modules found by WGCNA. But unlike WGCNA modules, they are trait dependent and exist at multiple overlapping levels. So we use the HOGs for various kinds of network interpretation. First, we perform functional enrichment analysis of KEGG and GO terms across all groupings and rank these terms both within the modules of each disease and across disorder. This slide shows across disorder rankings. RNA processing, synaptic, and immune-related pathways appear in many of the terms towards the top of this ranking, in agreement with independent analyses of the PsychENCODE data in the across disorder capstone paper. So we further visualize best paths through the network associated with several prioritized co-expression modules, showing that we are able to link key genes, SNPs, enhancers, and pathways for each disease. For example, in schizophrenia, the left panel links a key gene, GRIN1, with glutamatergic synapse functions and excitatory neuron cell types, while the right panel links genes in the complement pathway with astrocyte cell type and calcium signaling annotations, in agreement with recent findings. So we can also do this for bipolar and age phenotypes. And these are various other kinds of interpretations we can get out of this scheme, looking at enrichment for GWAS SNPs and high-confidence schizophrenia genes. So in summary, the PsychENCODE consortium has generated multi-omics data on a large scale relating to multiple psychiatric conditions and population-level variation. In order to leverage this data for de novo investigation of molecular mechanisms underlying these disorders, we propose an integrated deep learning framework which combines generative and predictive model architectures, incorporates latent factors, and allows prior structural knowledge to be embedded. Finally, we show how multi-level network interpretation methods can be developed to interpret latent factors as higher order groupings and prioritize genes and variants. So I'd like to conclude by acknowledging the NIMH and all of our collaborators in the PsychENCODE consortium. Thank you for your attention.
Thank you. Intriguing work. So I'm especially interested in the role of the interaction between the genes, the expression networks. I mean, you have cell-specific networks. What is the difference between compared to just a list of specific genes for cell types versus having interactions with the elastic net? What difference does it make?
So the cell type specific networks are actually found as subsets of a global genetic regulatory network. So we don't actually find cell type specific interactions. Within the deep learning framework, we have cell type variables in this network. So it would be possible to gain some of that interaction through those variables at that level. Yes, those networks that I showed were subsets of the global network. So yes, they're not actually cell, but except in the sense that those interactions will always be between transcription factors which are active in that particular cell type. So yes, it's I don't know.